Hello dear students, welcome to another tutorial from Brilliant Bios. In this video lecture we will learn about complex 2 of electron transport chain, and in the end of this video I will talk about another type of protein that actually functions very similar like complex 2. So in my previous video I have already discussed about complex 1, that is NADH CoQ reductase, how this complex functions and the structure of this complex. Now in this video I will talk about the complex 2. The complex 2, the name of this complex, the complex 2, let's suppose this is the complex 2 enzyme. So this complex 2 enzyme is known as succinate CoQ reductase. As I have mentioned, the name of this complex 1, that means NADH. So here NADH is actually oxidized and CoQ gets reduced, so this is called NADH CoQ reductase. So the name of this complex 2 also suggests the function of this complex, like succinate CoQ reductase. That means here succinate will get oxidized and CoQ, this means the ubiquinone, will get reduced. So what happens here? Okay, I want to mention the number of subunits. So this complex 2 actually composed of 4 subunits, and among them one subunit is very important. One subunit, that is the name of the subunit, is actually an enzyme called succinate dehydrogenase. Succinate dehydrogenase. So this complex 2, the succinate CoQ reductase, has 4 subunits. Among them one subunit is called succinate dehydrogenase, and I have already discussed about the function of this enzyme. The function of this enzyme is to convert one product called succinate to fumarate, and this reaction is reversible. So here also this enzyme is actually the part of this complex 2. So now you can understand the TCA cycle, because I have mentioned how succinate and fumarate gets converted in the TCA cycle. So this enzyme is actually part of the complex 2, that means it's part of the electron transport chain. So TCA cycle is physically and chemically linked with electron transport chain. Okay, now succinate to fumarate, this is the fumarate. Okay, so here is succinate molecule will be oxidized to fumarate. This reaction step is reversible, so I'm showing reversible step, fumarate. So you have to understand this. So this complex 2 contains a subunit called succinate dehydrogenase, is an enzyme that will convert this succinate into fumarate, plus 2 hydrogen atoms are actually released, protons are released. Now the high energy electron that are actually released by these reactions are actually taken by a cofactor or coenzyme called FAD. This FAD molecules receive 2 high energy electrons, and along with 2 protons it gets actually reduced to FADH2. So FAD gets FADH2. Then there is iron sulfur clusters present. So these are iron sulfur cluster. Okay, so what happens when succinate get reduced oxidized to fumarate? It releases 2 electron. FAD accept these 2 electron and actually becomes FADH2. Then other ferrous sulfur cluster actually carry the electron transport. As I have mentioned in this complex 1, that this direction of the electron flow is actually unidirectional, that means electron flow only in this way. For that reason the acceptor of this electron or the transporter of this electron should have low to high reduction potentials. I have already discussed about in my complex 2. So all these molecules should have higher redox potential. That means from succinate to fumarate, these reactions, this succinate and this FAD, this FAD should have higher redox potential so that it can receive this electron. Then this each and every ferrous cluster should have higher redox potential so that it can actually take the electron. Now in the matrix position, that means let's suppose here the co-Q actually is present. This is the co-Q, means ubiquinone is present. Ultimately electron move from this complex to here and jump from the electron ferrous sulfur cluster to ultimate to the co-Q, means ubiquinone. Ubiquinone can take 2 electrons and associate with 2 protons and becomes co-QH2. Becomes co-QH2. So let's suppose this is a co-QH2. Now you can see in case of complex 1, the ultimate product that actually gets reduced is co-QH2. So it's called co-Q reductase. In case of complex 2, there is also co-Q gets reduced to is called co-Q reductase. So that this is the ultimate product. Now the fact about this complex 2 is that this complex cannot transport or pump protons. They cannot, complex 2 cannot pump protons. So they do not actually directly take part in the proton motive force. But the delta J0 bar of this reaction is actually negative. But the energy that is released is insufficient for this high proton pumping. Okay, now I will talk about another protein set that actually remains actually present in the matrix region of this mitochondria and functions very similar like the complex 2. How it occurs, I will tell you. In the mitochondrial matrix, one important thing occurs that is called fatty acyl CoA oxidation. Means fatty acid oxidation. This is very important because fatty acids have so many carbon molecules and energy. So this fatty acyl CoA oxidation, this is also known as beta oxidations, because during each and every cycles these oxidations have 4 steps. This oxidation of these fatty acids has 4 steps, and during each cycle there is 4 steps. 2 carbons are actually released from the beta position, so that's why it is called beta oxidation. I will take a separate video or class on this beta oxidation of fatty acids. But the important fact about this mechanism in the electron transportation is here. In this 4th step, the first step of this beta oxidation, an enzyme actually take part that is called fatty acyl CoA dehydrogenase. You have to understand this. There are 4 enzymes, so there are 4 steps during these fatty acyl oxidations, also known as beta oxidations. But the first step is performed by an enzyme that is present in the matrix. So let's suppose this is the fatty acyl CoA dehydrogenase. So I am writing it FACD. So FACD. I am just giving the name FACD. So this enzyme can convert, actually helps in the dehydrogenations. I know I have already discussed about what is dehydrogenations, because dehydrogenase enzyme means it can exchange hydrogens. So this enzyme actually remove 2 hydrogens from C2 to C3 bonds, carbon bonds. It removes 2 hydrogens, and eventually this enzyme is actually convert FAD that is present inside this enzyme, one FAD to FADH2. So this enzyme, this fatty acyl CoA dehydrogenase, can reduce FAD and actually transfer 2 high energy electrons into the FAD. Now from the FAD, one molecule present in the matrix called ETF, this FAD is to release the high energy electron like this complex, FAD releases the high energy electron to the ferrous sulfur cluster, but here the FADH2 releases its electron into a protein called ETF. The full form of ETF is, remember, electron transfer flavoprotein. So FAD gets oxidized. FAD actually at first reduced, because this enzyme actually reduce, you have to understand this fatty acyl CoA gets actually oxidized by this, or dehydrogenased by this enzyme. So it actually remove two hydrogen atom from this C2C3 bonds, and it delivered these two protons and two electrons to the FAD that is present inside this enzyme. Now this FADH2 transfer these two high energy electron to a ETF, that is called electron transfer flavoprotein, that is present in the matrix, and ultimately it becomes into FAD again. Okay, now this E2F can reach into the inner membrane of the mitochondria, because here is a protein that helps in this pathway. Let's suppose this is the protein, it is called ETF QO. You have to understand this. So ultimately complex 1 make CoQH2, complex 2 make CoQH2, and the same thing actually occurs by this ETF QO. The full form of ETF QO is ETF QO. The full form of ETF means electron transfer flavoprotein. QO means ubiquinone oxidoreductase. Okay, ubiquinone oxidoreductase. So ultimately ETF transfer the high energy electron into this ETF QO, where the same coenzyme ubiquinone is actually present. So this is the CoQ. It receives this electron. And become reduced to CoQH2. So now you can understand this complex 1, complex 2 and this ETF QO, that is actually take part in the fatty acid oxidations, and ultimately it actually helps in the actually relate this mechanism with electron transport chain by reducing CoQH2O, and actually this CoQH2 has two high energy electrons. So the ultimate product of these three complex is the same CoQH2 that actually reach to the complex 3. So in our next class we will learn about complex 3 in more details, how the function and structure of this complex 3. Okay, so thank you for this watching.